Hi, my name is Isabelle Enzyme. This is our enzyme lab with Group D, Alex, Roya, Dishka, and myself. We studied the enzyme trypsin in this experiment. So, naturally, trypsin is an enzyme, and enzymes are protein catalysts that increase the rate of reactions, and they belong to the protein group of macromolecules, and they speed up reactions by lowering the activation rate needed. Enzymes are important because they cause reactions to speed up, and they're essential for human life, and that would normally not occur fast enough. The reaction trypsin catalyzed was broken down into azocasein in this experiment. The role of trypsin in an organism is that they digest dietary proteins in the small intestine. Trypsin is active in the conditions of areas in the body are in the small intestine, and they help digest dietary protein. And they're normally found in the neutral pH level of the small intestine. So for our experiment, our question and hypothesis was, how does the temperature affect the function of an enzyme? And our hypothesis was, if the sample is placed in a warmer temperature condition, then the enzyme will have an optimal performance. Our rationale for this hypothesis was that enzymes have an optimum performance at high temperatures because they increase the activity of enzymes and the rate of reactions until they further denature and then can't function no more. So the steps of our experiment included all the test tubes had to be labeled with different temperatures, so we decided to use 37, 50 degrees Celsius, 37 degrees Celsius, 50 degrees Celsius, and 60 degrees Celsius. The tubes were filled with 450 milliliters of reaction buffer, 500 milliliters of azocasein, and 50 milliliters of trypsin, and each tube was placed in a different water bath of 37 degrees, 50 degrees Celsius, and 60 degrees Celsius. And the buffers were allowed 10 minutes to incubate. Then we took the test tubes out, added 500 milliliters of 10% TCA caution in each test tube in order to stop the reaction from precipitating. And then in our next step, we centrifuged each test tube at maximum speed for 3 minutes. After 3 minutes, we transferred the supernatant to clean the cuvette and blanked the spectrophotometer at 440 nanometers with a cuvette with water. And then in our next step, we recorded the absorbances of our experimental groups, and once the spectrophotometer read at 440 nanometers. So the reason why we added TCA was that we had to stop the reaction of precipitation of the macromolecule proteins, which is what I said before. So why did we centrifuge the TCA and what was in the pellet? The TCA was centrifuged because we had to separate the larger particles from the solution, and the pellet consisted of the enzyme and the substrate. Azocasein was used as substrate, and sulfamilamide correlated to the activity of trypsin. Our control conditions were the pH and the salinity. So the pH was maintained at a level of 9, and the salinity was measured at a molar capacity of 0.15. And we used the pH level of 9 because other research concluded that enzymes performed better at a pH level of 9. So we also had negative controls prepared in our experiment, and we measured 2 sets for each. The negative controls prepped were the same way as the experimental groups, but we used water instead of trypsin. So we had 3 replicates, and we did this to reduce error and find the outliers in order to measure variability. Okay, so for our results, the average of the 62 degrees Celsius tube was 0.209, and the variability was 0.011. For 50 degrees Celsius, the average was 0.59, and the variability was 0.088. For 37 degrees Celsius, the average was 0.384, and the variability was 0.074. So, for this experiment, we had a p-value, which was also known as our t-test. Our t-test results showed that the absorbance at 50 degrees and 37 degrees and absorbance at 50 degrees Celsius and 62 degrees are significantly different. Our key results and variability were that there was a significant difference in absorbance and temperature between 50 degrees and 37 degrees with a p-value of 0.036 and between 50 degrees and 62 degrees with a p-value of 0.001 because if the p-value is less than 0.05, the difference in significance, so that we can conclude from our result that the null hypothesis is supported, and 5%, which was 5% confidence, and the alternative hypothesis is supported by 95% confidence. Our experimental design was adequate to test our hypothesis. However, our execution was not. We had errors mainly with temperature of the water bath and the specimen. Our hypothesis was partially supported in the experiment because they showed that the temperature at 50 degrees Celsius is the temperature that trypsin had an optimum performance at. Compared to the other groups, we were the only group that used temperature as our experimental variable, but we used a pH level of 9 and salt concentration of 0.15 molar because that's what other groups tested and said what was the optimum for each experimental variable for trypsin. I think that what we did for our experiment was the best outcome because our choice of control variable conditions resulted in the highest possible rate of enzyme activity. We maintained a pH level of 9 because 3 groups tested pH and the optimum level was 9 because trypsin had a better performance at a higher pH that didn't denature it in harsh conditions. A major source of variability we had was the inconsistency of our water baths. This was problematic because then all the trials were not held at the same temperature, which resulted in the fluctuation of temperature and the absorbances. So to improve our experiment from the first to the second attempt, we increased our pH from 8 to 9 and increased our experimental temperatures, and these changes allowed us to have more ideal conditions. For future experiments, it would be beneficial to make improvements to prevent error in our water baths. So the ideal conditions of bacteria are normally warmer temperatures because that is what they thrive in. Warm temperatures increases the rate of growth and helps bacteria to maintain homeostasis in order to survive and grow.